You can eat less than you did 20 years ago, genuinely less, smaller portions, no dessert, coffee instead of a second helping, and your belly still grows. Not slowly, not negligibly, stubbornly, persistently, in a way that makes no sense on any calorie spreadsheet you have ever looked at. That is not a willpower failure. That is not age being unkind in a vague, shruggable way. That is a set of four overlapping biological mechanisms that are actively routing fat to the deepest, most dangerous location in your body right now, and calorie cutting alone cannot touch a single one of them. By the end of this, I am going to give you something called the Depot Shift Protocol. Three specific interventions that target the exact biological layers responsible for post 55 belly fat. I will name it now because I want you to see, as we work through each layer, precisely why each piece of that protocol exists and what it is actually correcting. This is not a list of tips, this is the mechanism, and the mechanism is what nobody has walked you through. I want to start with something that changed how I thought about all of this. My husband, about 8 years ago, post-aneurysm, handed a prescription stack and told to take it easy, kept asking why his belly was growing when he was eating barely anything. His doctor said his numbers were improving. His waist kept expanding. I started reading. What I found was not complicated in isolation, but it was layered, and every layer was hiding something the one above it had concealed. So let us peel them. The first thing to establish is what belly fat actually is, because the mainstream conversation treats it as a single thing, and it is not. When most people say belly fat, they mean the soft, pinchable layer just under the skin. That is subcutaneous fat. It is aesthetically annoying. It affects how clothing fits. But from a metabolic standpoint, it is relatively inert. It stores energy, it insulates, it does its job quietly. Then there is the other kind, the kind you cannot pinch because you cannot reach it. The kind sitting deep inside your abdominal cavity, wrapped around your liver, your pancreas, your intestines, your kidneys. That is visceral fat. And visceral fat is not storage, it is a functioning endocrine organ. It produces hormones. It releases inflammatory proteins called cytokines, signaling molecules that travel through the bloodstream and alter how your heart, your liver, your immune system, and your brain operate. It communicates directly with your liver in ways that degrade your body's ability to process glucose and regulate cholesterol. Here is the metaphor I want you to hold throughout this entire video. Your body is a warehouse with two storage zones. Zone A is the regular warehouse floor, accessible, organized, used for normal short-term inventory. That is your subcutaneous fat. Zone B is a locked back room. Inventory that goes in there is difficult to retrieve, and while it sits there, it is not passive. It is actively sending signals into the warehouse management system, corrupting data, overriding instructions, making the entire operation progressively less efficient. After 55, something changes in the shipping algorithm. More and more of what enters the building gets routed directly to Zone B. Even when you reduce total incoming shipments, eating less, cutting portions, the routing algorithm keeps sending a disproportionate percentage to the back room. The problem is not the volume of inventory. The problem is where it goes. And what controls the routing is exactly what we are about to strip away layer by layer. The first layer is hormones. Specifically, what estrogen does for fat distribution in women, and what testosterone does in men, and what happens when those hormones decline. Most people know estrogen drops during menopause. What most people do not know is what estrogen was actually doing for fat storage routing in the first place. Estrogen, at premenopausal levels, actively suppresses visceral fat accumulation. It does this by influencing alpha and beta adrenergic receptors, the receptors sitting on the surface of fat cells that determine whether the cell opens to release stored fat or closes to hold it in. Estrogen essentially programs fat cells in the hips, thighs, and gluteal region to be the primary storage sites by keeping the abdominal fat cells in a relatively closed, release-ready state. It is not random that younger women carry more fat in those peripheral areas. It is a specific biological instruction encoded by estrogen's interaction with fat cell receptors. When estrogen drops not just at menopause but during the years of perimenopause before it, that instruction disappears. Abdominal fat cells, which carry more of the receptor subtype that responds to storage signals, suddenly become the dominant depot. The body does not stop storing fat. It changes the zip code. A 2012 study, published in the journal Menopause, tracked changes in body fat distribution across the menopausal transition and found that visceral fat increased by an average of 49% over a 5-year period, even in women whose total body weight changed relatively little. The belly grew not because those women ate more. It grew because the biological routing instruction had been revoked. For men, the mechanism runs in parallel, but through a different molecular pathway. Testosterone, which declines at roughly 1 to 2% per year after 30 and drops more steeply after 50, normally suppresses an enzyme called lipoprotein lipase in abdominal fat cells. Lipoprotein lipase is essentially a fat absorption enzyme. Its job is to pull triglycerides out of the bloodstream and drag them into fat cells for storage. When testosterone is present at adequate levels, it keeps this enzyme relatively quiet in the belly region. As testosterone falls, that suppression lifts. Abdominal fat cells become increasingly aggressive at capturing circulating fat from the bloodstream. The more testosterone has dropped, the more voracious the belly fat cells become at pulling in everything that passes by. In both men and women, the first layer of the problem is this. The hormonal programming that told your body where to store fat has been quietly overwritten. Not because you changed your diet, not because you stopped exercising, because the instructions that governed fat distribution for 30 years simply expired. The second layer goes deeper, and it involves something that does not get nearly enough attention when people talk about belly fat after 50, muscle. Adults lose somewhere between 3 and 8% of muscle mass per decade, starting around age 30. After 60, that rate can double. This has a name, sarcopenia, and it is one of the most underestimated drivers of what people describe as a slowing metabolism. Here is why muscle loss matters so directly. Resting muscle tissue burns approximately 6 calories per pound per day. Fat tissue burns roughly 2 calories per pound per day. Across an entire body over years, that difference accumulates into something enormous. A person who loses 10 pounds of muscle between 40 and 55, which is entirely within the normal range for someone who does not specifically work against sarcopenia, has reduced their resting daily calorie expenditure by roughly 60 calories per day. That sounds modest. Over a year, it equals about 22,000 calories. That is approximately 6 pounds of potential fat accumulation per year, derived entirely from muscle loss, even if eating habits have not changed by a single calorie. But here is the piece that gets almost no coverage in mainstream health advice, and it is the one that causes the most damage. When you eat less to compensate for a seemingly slower metabolism, you can inadvertently accelerate sarcopenia. Caloric restriction without adequate protein intake triggers your body to break down muscle tissue to use as fuel. The very strategy most people reach for, portion control, eating less, can reduce the calorie-burning machinery that would have been doing the metabolic work. This is the warehouse running with a shrinking loading dock. The operational capacity has already been reduced by the muscle loss, and now you are also reducing incoming inventory, but the routing algorithm is still sending a disproportionate percentage straight to Zone B. Now layer insulin resistance on top of this, and the second layer becomes much more consequential. Insulin is released by your pancreas every time you eat carbohydrates, and to a lesser degree protein. Its job is to open cellular doors and let glucose in. Muscle cells are the primary site for that glucose uptake. They absorb the majority of what you eat when they are adequately present and sensitive to insulin. When muscle mass decreases, there are fewer doors. Glucose that cannot get into cells stays in the bloodstream. The pancreas responds by producing more insulin, trying to force the doors open with a stronger signal. Chronically elevated insulin has one critically relevant effect. It locks fat cells shut. When insulin is high, the enzyme inside fat cells that releases stored fat, hormone-sensitive lipase, is suppressed. Fat cannot exit the cell while insulin is elevated. In a body with reduced muscle mass, insulin stays elevated longer after every meal, not because the meal was unusually large, but because the glucose uptake capacity has been degraded. During those extended windows of insulin elevation, you are biochemically blocked from burning stored fat for fuel. You are locked out of your own fuel reserve. This is not a metaphor. This is happening in your body right now. If you are over 55 and have not deliberately worked to maintain muscle mass through resistance training, the door count is low. Insulin stays elevated after meals for longer than it did at 35. Fat cells stay locked. New fuel arriving from food gets routed to zone B. Here is where I need to stop and address something that has done more damage to people in this age group than almost any other single piece of guidance in mainstream health. I say this not with anger. I genuinely find it sort of fascinatingly absurd in hindsight, but the calories in, calories out model, applied without modification to bodies over 55, produces outcomes that contradict its own stated goals. We have been told for decades, eat less, move more, calorie deficit equals fat loss. The formula sounds mechanically airtight. A body of research accumulated over the past 20 years demonstrates that it is dangerously incomplete when applied to postmenopausal women and men over 55, and in some cases, it actively worsens the problem it claims to solve. Here is the mechanism that the calorie model ignores entirely. When you significantly reduce caloric intake, particularly without adequate protein and without resistance training, your body interprets that restriction as a threat signal, not as an invitation to draw on stored fat, as a sign that something is wrong. The hypothalamus, the region of the brain that manages survival responses, detects the deficit and activates the stress axis. The result is a rise in cortisol. Cortisol is your primary stress hormone, produced by the adrenal glands sitting above your kidneys. In acute situations, running from a threat, managing a short-term crisis, cortisol is enormously useful. It mobilizes energy rapidly. It sharpens focus. It is designed for emergencies that last hours, not months. Chronically elevated cortisol, the kind sustained over weeks or months of ongoing restriction or life stress or poor sleep, has a specific and repeatedly documented effect on fat distribution. Cortisol preferentially drives fat storage to the visceral depot. The reason is molecular, visceral fat cells have significantly higher concentrations of glucocorticoid receptors, the cellular receptors that bind to cortisol, and execute its instructions compared to subcutaneous fat cells. When cortisol is chronically elevated, it is broadcasting a direct instruction: fill zone B. Dr. Alyssa Appel and colleagues at Yale University published a series of studies in the early 2000s demonstrating that cortisol reactivity predicted visceral fat accumulation independently of total calorie intake. The finding was replicated across multiple populations. The belly was not filling because of calories. The belly was filling because of cortisol. Let that sit for a moment. Independently of total calorie intake, you could be running a genuine caloric deficit. Your food diary could be accurate, and the math could be correct. And cortisol, elevated by the restriction itself, by poor sleep, by daily stress, by the physiological stress of hormonal decline, could be systematically instructing your body to store fat in the deepest, most dangerous, most metabolically disruptive location. The prescription to eat less can trigger the exact mechanism that makes the belly grow. This is not a niche or fringe finding. This is repeated across multiple peer-reviewed bodies of work, sitting in plain sight in academic literature, and not once mentioned in the 15-minute appointment where someone is told to cut calories and exercise more. I do not find this enraging anymore. I find it a remarkable illustration of how wrong an incentive structure can get, and how long it can stay wrong. Before the next layer, I want to mention something briefly. Knowing all of this and having a structure that actually implements it are two different things. I had read the studies on circadian eating windows, on protein distribution, on the cortisol restriction trap. I had bookmarked the research. I was still not doing it consistently because understanding a mechanism and building a daily habit around it are completely separate cognitive challenges. My husband spent the better part of a year building something out of all of this, not because he is particularly methodical by nature, but because after nearly dying, methodical started looking attractive. He called it the Noon Reset Protocol. It is a 30-day daily companion. 2 pages per day, 1 page of the science for that day so you understand what you are actually doing and why it works at this specific stage, and 1 page of tracking so you can see the changes accumulating. He built it around 4 phases: adaptation, activation, optimization, and acceleration, because anyone who has tried to change eating habits knows that day 4 is a completely different challenge than day 14, and the structure needs to acknowledge that. He also built out 25 breakfast meal ideas with exact protein counts, because the first meal of the window is where most people either hit the threshold that triggers muscle protein synthesis or miss it and wonder why they are not making progress. You can find it through the link in the description. It costs less than dinner out. And now, the third layer, because this one reshapes everything we have discussed so far. The third layer involves a hormone most people have never heard of, and it is arguably the most elegant piece of the entire system because it demonstrates how visceral fat does not just create problems, it perpetuates them. That hormone is adiponectin. Adiponectin is produced by fat cells, specifically by subcutaneous fat cells in healthy amounts. It circulates in the bloodstream and performs several protective functions simultaneously. It improves insulin sensitivity, which means it helps muscle cells and liver cells respond better to insulin's signal and take up glucose more efficiently. It suppresses inflammatory pathways, acting as a brake on systemic inflammation. It inhibits the accumulation of fat inside the liver, a process called hepatic steatosis or fatty liver disease, by directly acting on liver cell metabolism. Research from multiple institutions has linked higher adiponectin levels to reduced risk of cardiovascular disease, type 2 diabetes, and what is now called metabolic syndrome. Here is the critical structural problem. Visceral fat cells produce very little adiponectin. And as visceral fat accumulates, it does not simply fail to contribute adiponectin, it actively suppresses the signal from nearby subcutaneous fat cells. Total adiponectin in the bloodstream falls as the visceral depot grows. Postmenopausal women, according to research published in the Journal of Clinical Endocrinology and Metabolism, show adiponectin levels roughly 20 to 30% lower than premenopausal women matched for body weight and height. Same person, functionally, same total fat mass, but the fat has redistributed internally, and the protective hormone output has dropped by nearly a third. Lower adiponectin means worse insulin sensitivity. Worse insulin sensitivity means higher and more prolonged insulin spikes after meals. Higher insulin means more fat storage, more cortisol signaling, and preferential routing to zone B. More zone B fat means less adiponectin. The cycle is not metaphorical. Each turn tightens the loop. The warehouse's backroom is not just filling, it is releasing chemicals that cause the loading dock to route more to the backroom. There is one additional mechanism here that most health content misses entirely, and it connects to liver function. Visceral fat sits in what anatomists call the portal drainage area, meaning the blood that circulates through visceral fat deposits drains directly into the portal vein, which feeds directly into the liver. Subcutaneous fat does not have this arrangement. Fat metabolites, inflammatory cytokines, and free fatty acids from visceral fat deposits go straight to the liver before they reach general circulation. This is why high visceral fat is so specifically linked to non-alcoholic fatty liver disease, why it elevates triglycerides so reliably, and why it impairs hepatic insulin sensitivity in ways that general obesity does not. The liver is receiving a steady chemical feed from the visceral depot at close range. It responds by becoming progressively less efficient at the metabolic tasks that would otherwise help manage body composition. The fourth layer descends to the cellular level, and it explains something that no amount of calorie adjustment has any mechanism to correct. Your fat cells contain mitochondria, the same energy conversion structures found in every cell in your body. Mitochondria take fat and carbohydrates, combine them with oxygen through a complex series of reactions, and produce ATP, the actual usable fuel that powers every biological process. Think of mitochondria as the combustion engines of each cell. Without functioning mitochondria, stored fuel cannot be burned, regardless of how much or how little is available. After 50, mitochondria age. Their number in fat cells decreases. Their efficiency, measured by how much ATP they produce per unit of oxygen consumed, declines. Damaged mitochondria accumulate and are cleared more slowly than in younger tissue. This is not speculative. It is one of the most robustly documented aspects of cellular aging. But here is what makes this specifically relevant to belly fat and not just general metabolic decline. A 2019 study from researchers at the Mayo Clinic, published in the journal Diabetes, demonstrated that visceral fat depots in older adults showed significantly higher rates of mitochondrial dysfunction than subcutaneous fat depots, meaning the internal combustion engines of visceral fat cells were running at much lower capacity than those in subcutaneous fat cells, even in the same person at the same time. Visceral fat had older, more damaged, less functional mitochondria, and lower mitochondrial function in fat cells means reduced fat oxidation, literally less ability to burn the fat sitting in those cells. This is why the belly is the last place to change and the hardest place to target. It is not stubbornness in any psychological sense. The cellular machinery that would burn that stored fat is running at reduced efficiency. You are trying to drain a tank with a pump that is operating at 30%. The remarkable corollary to this, and this is what made the protocol make sense to me biologically, is that mitochondrial biogenesis, the creation of new mitochondria, is one of the few aging-related cellular declines that responds meaningfully to a specific, well-characterized behavioral intervention. That intervention is sustained moderate-intensity aerobic exercise, not sprints, not weight training, though that has its own role. Sustained aerobic work at the intensity where you can speak but would not choose to activates a master regulatory protein called PGC1 alpha, which triggers the creation of new mitochondria in muscle and fat tissue. The old engines do not disappear, but new ones are added alongside them. The overall capacity of the visceral fat tissue to actually burn its stored fuel increases over weeks and months of consistent aerobic activity. This is one of the few places where the biology offers a direct repair pathway, not just damage control. The final layer ties every previous one together and explains why the timing of food intake matters as much as, and in some contexts, more than, what you actually eat. Your body runs on a 24-hour internal biological clock called the circadian system. Every organ has its own peripheral clock synchronized to the master clock in the brain by two primary signals: light and the timing of meals. Your liver has one, your pancreas has one, your fat cells have one, your intestinal cells have one. These clocks collectively determine what your body does at which hour of the day, when it prioritizes fat oxidation versus fat storage, when insulin sensitivity peaks and when it troughs, when growth hormone is released and when it is suppressed, when the gut microbiome produces which metabolites. In the morning and early afternoon, insulin sensitivity is at its physiological peak. The same carbohydrate load eaten at 9 in the morning produces a smaller, shorter insulin spike than the identical meal eaten at 8 in the evening. The pancreas issues less insulin. The cells respond to it more efficiently. The window of elevated insulin is shorter. The period during which fat cells are locked by insulin is briefer. More of the day's metabolic activity is spent in a state permissive to fat oxidation. After 55, this circadian disparity between morning and evening insulin sensitivity becomes more pronounced, not less. Research from the University of Aberdeen and replicated in subsequent studies has demonstrated that older adults show a markedly greater glucose excursion from an identical evening meal compared to the same meal consumed in the morning, a difference considerably smaller in younger adults. The aging circadian system amplifies the metabolic penalty for evening eating. This matters enormously because the default eating pattern in most developed countries runs in exactly the wrong direction. The morning meal, if consumed at all, is typically the smallest. The evening meal is typically the largest, often consumed between 6 and 9 at night. This is the biological worst-case scenario for someone over 55 with declining hormones, reduced muscle mass, elevated cortisol, and declining adiponectin. The largest fuel delivery arrives at the moment when the warehouse is least equipped to process it efficiently. The routing algorithm sends as much as possible to Zone B. Insulin stays elevated deep into the night, fat oxidation is suppressed for hours, and the mitochondria in visceral fat cells, already running at reduced capacity, are not called upon to do any burning during those overnight hours because insulin has locked the cells shut through the early morning. Now bring all 5 layers together simultaneously, because this is what is actually happening in a body over 55. Estrogen or testosterone has removed the hormonal programming that kept fat out of the visceral depot. Sarcopenia has reduced glucose uptake capacity, keeping insulin elevated longer after every meal. Cortisol, possibly elevated by chronic undereating, by poor sleep, by stress, or by all three, is sending direct instructions to fill Zone B. Adiponectin has fallen, worsening insulin sensitivity and creating a reinforcing loop. Mitochondria and visceral fat cells are too aged and dysfunctional to burn what is being stored there efficiently. And the largest meal of the day is arriving at the worst possible circadian moment for metabolic processing. No calorie count addresses any of those 5 layers. The number on the food diary is correct. The biology is simply not organized around that number. Metabolic is where the depot shift protocol exists. 3 interventions, each targeting a distinct layer of this system. The first component is the compression window. This is time-restricted eating, anchored specifically to your body's circadian insulin sensitivity curve. The goal is to complete all eating before insulin sensitivity begins its evening decline, which for most adults over 55 means the eating window closes by 6 or 7 in the evening at the latest. The research supporting this is not about starvation. A 2020 study published in Cell Metabolism by researchers at the Salk Institute demonstrated that time-restricted eating, without any intentional caloric restriction, reduced visceral fat specifically, not just total body fat, but the internal visceral depot, in adults with metabolic syndrome. The mechanism was precisely the circadian layer we just discussed. Less evening insulin, more overnight hours in a state permissive to fat mobilization. The window I use personally starts at noon. That creates a 6-hour eating window during the circadian period of highest insulin sensitivity and a 17-hour window during which the body, with insulin low, growth hormone cycling, and cellular repair processes active, can actually work on what is stored in Zone B. The 2 eating occasions within that window are noon and early to mid-afternoon, with the larger of the 2 meals consumed at the earlier time slot, not the later one. The second component is priority protein loading. This addresses the sarcopenia layer directly by ensuring that each meal within the eating window reaches the specific protein threshold required to stimulate muscle protein synthesis in adults over 50. Research from the University of Texas Medical Branch led by Dr. Douglas Patton-Jones established that muscle protein synthesis after 50 requires not just adequate total daily protein, the number often cited, around 0.8 grams per kilogram of body weight, but a specific dose per individual meal, approximately 30 to 40 grams of protein per eating occasion, to maximally stimulate the repair and maintenance signal. Most people who eat enough protein over the course of a day distribute it poorly. Very little at the first meal, moderate in the middle, heavy at dinner. 2 out of 3 meals miss the stimulation threshold entirely. Priority protein loading means that at noon, when the fast is broken, the first thing consumed is a meal centered on at least 30 grams of complete protein. At the second meal, the same target is hit again. The specific sources that are most effective for this purpose are leucine-rich proteins, animal proteins, particularly eggs, fish, poultry, and dairy, or for plant-based eaters, combinations that ensure adequate leucine delivery, such as legumes alongside a whole grain source. Leucine is the specific amino acid that acts as the trigger for the muscle protein synthesis pathway, and older muscle tissue requires more of it per meal than younger tissue to generate the same anabolic response. The third component is the Zone B drain. This is the aerobic exercise prescription, and it is considerably more specific than any general advice to be more active. Visceral fat, Zone B responds preferentially to aerobic exercise above almost any other intervention, including caloric restriction. A meta-analysis published in Obesity Reviews examining over a dozen clinical trials found that aerobic exercise reduced visceral fat area by an average of 6.1%, even in trials where participants showed minimal change in total body weight. The mechanism is threefold. Aerobic exercise transiently suppresses insulin and activates fat mobilization pathways, directly stimulates PGC1 alpha, and triggers mitochondrial biogenesis in both muscle and visceral fat tissue, and creates a post-exercise window of reduced insulin sensitivity in fat cells that extends fat oxidation for hours after the session ends. The specific prescription from the accumulated evidence is moderate intensity aerobic work, the zone where you can speak a sentence but would not choose to do so comfortably, sustained for a minimum of 30 minutes per session, at least 5 days per week. That is 150 minutes per week of moderate aerobic work as the floor. The visceral fat response is dose-dependent up to approximately 150 to 170 minutes weekly, after which the curve levels off meaningfully. A 20-minute walk twice a week produces a fraction of the visceral fat response of a 30-minute daily walk. The duration and frequency are not arbitrary numbers. They are the inputs at which the mechanism actually engages at a clinically meaningful level. The single most common mistake that neutralizes this protocol is implementing it on top of severe caloric restriction. The compression window is not a starvation protocol. Time-restricted eating works because of the timing signal it sends to the circadian system, not because it forces a deficit. Adding a severe caloric deficit activates the cortisol pathway, reinstates the zone B routing signal, and undoes the work the aerobic component is trying to accomplish. If your daily intake is falling below roughly 1,400 to 1,500 calories while following this protocol, the cortisol elevation is likely actively working against the visceral fat reduction. The intake needs to be adequate, distributed correctly within the window, and centered on hitting protein thresholds, not minimized. The reason most people over 55 cycle through the same experience, restrict, lose a little, plateau, regain with extra belly fat, restrict again, is not discipline. It is not moral failure. It is having a model of the system that was accurate in a younger body and has not been updated to account for 5 simultaneous biological changes happening in parallel. When the map is wrong, you get lost even when you are walking confidently and quickly. The mainstream instruction, eat less, move more, was never a complete model even for younger adults. Applied to a post-55 body with hormonal redistribution, sarcopenia, insulin-cortisol dynamics, declining adiponectin, aging mitochondria, and compressed circadian insulin sensitivity, it becomes actively counterproductive in many cases. The mechanisms we have walked through are not obscure. They are in peer-reviewed literature. They are in the work of researchers at the Mayo Clinic, Yale, the Salk Institute, the University of Texas Medical Branch. They are not alternative health claims. They are mainstream biology that somehow never made it into the mainstream health conversation. My best guess at why, and I say this with more amusement than outrage at this point, is that the conversation that would explain all of this correctly takes about 40 minutes and requires a model of hormones, mitochondria, circadian biology, and protein distribution that does not fit neatly onto a pamphlet or into the standard appointment. Complexity is expensive. Simplicity sells. Eat less is simple. The mechanism of visceral fat accumulation in a postmenopausal body, navigating cortisol, sarcopenia, and circadian insulin sensitivity is not. So we got the version that was easy to say, even though it was not fully true. You now have the version that is actually true. The layers are clear. The mechanisms are specific. The interventions map directly to the biology. If you are starting the Depot Shift protocol this week, comment Zone B below so I can see who is actually doing this. That is the only metric that matters to me. Not views, not likes, not algorithmic favor. Who is actually moving from watching to implementing. And if this is the first video of mine you have come across, the subscribe button is right there. I read the studies, the clinical guidelines, the meta-analyses, the industry reports, the things most people would rather not read, and then I tell you what actually matters from all of it.